about algorithms. Algorithms in the whole life are usually boring, but sometimes necessary. And algorithm, in my case, has a bigger ambition to contribute for the evaluation of tumor markers for diagnosis and therapy monitoring in cancer diseases. So what is the background? Very shortly, in our project, biomarkers are used in daily clinical practice, particularly for the recurrence detection during the follow-up period of the cancer diseases. They are rarely used for the diagnostic purpose or therapy monitoring. Now, the aim of our study, of course, because not only me, was to propose algorithms for optimization of the diagnostic approach for the cancer diseases and for the choice of the optimal therapy and for monitoring of therapy effect based on serum levels of tumor markers. It means that we will speak about another family of tumor markers as before. So, our study is based on retrospective evaluation of 30,000 results of serum tumor markers from 2,000 patients, only for the region around Pilsen, and during the period of 10 years. And what cancers were chosen? We evaluated breast, colorectal, prostate cancer, and lung cancer. And the following markers were used. It means that they are from different families, and I will later explain you why. So I will answer the question you will ask me first. Why we decided these cancers and why these biomarkers? And the answer is very simple because it's a retrospective study. So we had information about this complete. So I will save your time. Then the next, results. So the results are concerning diagnostic algorithms in the tumor of unknown origin, differential diagnosis, early diagnosis, follow-up and disease course monitoring, therapy effect monitoring for surgery, adjuvant or palliative therapy, chemotherapy, and prognosis estimation. So, cancers of unknown origin. So we know that cancer is not a simple disease, so we usually meet patients suffering from different and combination of diseases. And we sometimes face the confusion of histology and immunohistochemistry. So the markers used here are named. So the first two markers, yes, are embryonic antigenes, cancer embryonic antigenes, and others are enzymes and also cytokinins. So we don't use them for primary diagnosis because of pure sensitivity and specificity, what we know, of course. Because in our inner Czech rules, when the biomarker can be used as prognostic, it must be, we have a very high sensitivity and specificity. And none of them used here can reach these levels. So now here we are with the first software which can help us to estimate what kind of diseases is it. If it is unknown time and location of primary tumor, when metastases are diagnosed or there is a high suspicion of cancer disease. So physician can add to this software, I have it in my laptop, how is it called, this small iPad, so it really exists, so you can find it, and it will help you to solve the problem where the metastasis is and primary tumor could be found. So in differential diagnosis, we can compare two illnesses, one malignant and one benign, and using combination of two biomarkers, we can reach 82% sensitivity and 95% specificity. But when we add another thymidine kinase, the sensitivity is 10% higher. The same is in another case, and we used for one or combination of three biomarkers. It is the same. result. Now, let me show you how we use this for optimization of early diagnosis in prostate cancer. So for us, this is prostate-specific antigen, and the cutoff value is 10. So this value is index of health prostate and is calculated on another factors, and the cutoff value is 40. So what does it mean? It means that using these two parameters, we can save biopsy for these two categories of patients and also magnetic resonance. In the numbers, when we calculated 1,350 patients, for biopsies we saved 200, sorry. For magnetic resonance also 200, and for this combination 150 patients. So it's, yes, so it saved not only patient and the doctors, but also money. Disease follow-up monitoring. So this is a comparison of levels of CEA. In these two cases, they are, yes, oh, no, the left two columns are benign, and the three right are colorectal, breast, and liver metastasis. You can see the levels, the blue levels of CA before operation. And then you can see the levels of the first, second, and after the 30 days. What it means, in the left two columns, the levels of CNA were very low before during the whole period. And the cutoff level for CA is 5%. So you can see that here it is under the level, and the 30 days in all cancers, the levels were at under the cutoff value. Another, sensitivity of thymidine kinase in colorectal cancer, and this shows us the period prior clinical manifestation. Oh, this one is better. Thank you. Yes, thank you. So 9 months before, the levels of all these biomarkers were very low. And after 3 months, they were increasing a little bit. And you can see that when we combined 2 biomarkers or 3 biomarkers, the sensitivity and specificity was higher. So using the combination of this biomarker showed us better and more precisely the preclinical manifestation. This is the same in the breast cancer. So in 356 women, also you can see that we evaluated 3 biomarkers, so 3 separately, and the last column is together. So again, it shows better results prior the clinical manifestation. And this is the last example of liver cancer. Again, the same results. So the second software is called Corrector, and it will estimate the prognosis of certain tumor diagnosis. So it will, when you ask this software, well, how it clinically evident metastasis develop in a case of a patient in a remission by the next checkup, it will help you to answer the risk of development of distant metastasis in the course of approximately next 3 months and their location. And the physician again can add additional information to make the prognosis and the answer from the software more precise. Now, therapy monitoring. This is a case of adjuvant chemotherapy in the cycle of 6 chemotherapy cycles. So the levels of thymidine kinases are not very high. Again, I mentioned that the cutoff is 10 in this case, so it's not. But after the chemotherapy, they grow up. It means that the chemotherapy was successful because, to make the long story short, there was blocked some synthesis around DNA, and the thymidine kinases had to grow up to support the normal life of the DNA and on the cell. So it is in the adjuvant chemotherapy. The next example is in palliative chemotherapy. So we started with the higher basic levels, but the shape of the line is the same. So also in the palliative chemotherapy, the thymidine kinase show the same results. Now in numbers, so this is successful palliative therapy, and you can see that these two additional biomarkers, which started in very high levels, were going down during, I don't know, 3 months, yes, rapidly. So for CA, as I mentioned, 10 is the right cutoff value, and in CA19 it is 40 or 50, so it's not as good result, but you can see that the tendency is very well. And this is example how when it doesn't work. So this is unfavorable prognosis for the patient in a palliative chemotherapy. And now here are results for C and CA19-9. You can see that the levels during only 2 months, but doesn't matter, are also the same. And so this therapy was unsuccessful. And now, conclusions. So we are in conclusions. So the optimal diagnostic algorithms were proposed for diagnosis and therapy monitoring for this mentioned 4 cancers. Selected cases reports demonstrated use of them and validity of them, and clinical and economical benefit of those proposed algorithms were evaluated. It means to save not only money, but also patient and the doctor. Next conclusion, a multidisciplinary approach based on these algorithms will enable to use the tumor markers for routine clinical practice much more effectively, not in the single case, but in the combination. Then perspectives for the methodology. It's mainly in multiparametric and multiplex assays with computer-assisted interpretation. We will use them in circulated tumor markers for the future, and they are also perspective in genomics, proteomics, and macroarrays. And as we mentioned here several times, personalized medicine in the approach, which it answers also these questions. For clinical perspective, the algorithms to individualization of target diagnosis and target therapy, correlation of biological activity of tumor with imaging techniques. Yes, this is new branch we are now focusing on, and incorporation of tumor markers into clinical and pharmacological trials. And this final goal is to prevent improvement in quality of life and better survival rates for our patients. Thank you very much. Hope I was